the first whenever the pathogen try to enter into the body, there's a kind of barrier. Our body it made some kind of barrier, mechanical barrier, for preventing the entry of the pathogen into the body, right? So as I discussed in the previous class, we have different types of microorganisms. And need not be microorganism, right? So just I'm taking different types of microorganism just for simplicity. So it can be a bacteria, or it can be a virus, or it can be a, in some cases it can be fungi or parasites. So many things, right? So can be anything, right? So the main job of this microorganism is that they have to somehow enter into the body. So because they have to access, they need some host for the replication or reproduction or the survival of the microorganism, right? So so if we consider this as the body, okay, so in the outer we have some kind of barrier, that what we call it as the mechanical barrier. Again, I cannot explain everything, right? So I'll share the video, right? So you may go through that, just some idea, right? So what I have discussed. So the very first mechanical barrier is the skin. So our skin is made up of different layers, multiple layers there in the skin, keratinocytes, right? So it's made up of different layer. So let's say, for example, the pathogen is present somewhere outside the body, which is not so happy. Okay, so the reason is that it is looking for some host, right? So without the host, the pathogens, it cannot be happy, right? So they're not so happy, right? So somehow this pathogen should enter into the body. So when the pathogen can be very happy, once if it successfully entered into the body, obviously it can use the machinery of the host and it can enjoy everything, right? So now the one which is present outside the body, absolutely it is not so happy, right? So it will always try to find some host. Okay, so we as a human, we are also host for the microorganisms, right? So whenever the microorganism, whenever the pathogen try to enter into the body, right, so our body put up the first kind of barrier, okay, mechanical barrier, called as skin. So the viruses or the bacteria, it should breach somehow, penetrate into the skin to get access inside the body. Otherwise, so that is the first kind of barrier provided by our body, right? Second one is, there are some places in which we cannot expect. Our body is not completely covered by skin, right? So we have to accept that. There are some part of the body which has some open access, right? Say for example, if you take mouth or if you take eye, right? So we cannot say that those are the organs which are protected by completely skin. So where the bacteria or the virus, it can try to enter through that region, okay, because skin is a big barrier for any of this pathogen. So they may try to access through that GI tract or through eye, okay, mouth, right? So the nature which made some kind of moist in those regions. If you take the GI tract or eye, right? So we have some kind of moisture, and if you carefully look at what are the components of those moisture, so those moisture, wet thing which contain different types of chemical compounds, different types of enzymes, right? So all those have kind of ability to stop the entry of this pathogens inside the body, and they have the ability to kill the bacteria or the viruses, right? So those molecules and enzymes which are present in the moist, they also act as a kind of a mechanical barrier. The pathogens are very clever, right? So if this entry is prevented, right, it will try to access through some other entry, right? So if we take lungs, or if you take pulmonary surfaces, so it has some free access for the incoming bacteria or the viruses, right? So nature, it made some kind of stuff even in the lungs, pulmonary surfaces called as cilia. Okay, so whenever the bacteria or the viruses, they try to access through the lungs or the pulmonary surface, because of the presence of this cilia, right? So what it does, the bacteria or the viruses which are trying to enter into the body to outside of the body. So that is another kind of mechanical barrier, right? So again, if we take as I said in the previous lecture, not all the bacteria, viruses, they do harm for the human system. There are some bacteria, viruses, we can say those are really a good bacteria, viruses, which are very important for the normal functioning of our body, right? So many gut bacteria there, right? So that what they call it as flora. Okay, bacterial flora, which means that they are really good bacteria, which are very essential. They really they really help the human system, right? In one or another way, right? So what it does, in the very surface, right? So we can find many number of different types of this flora, good bacteria or viruses or any other microorganism. They made a kind of coating like thing, right? So they occupy the surface. So which means that okay, there are many so let me illustrate this in a different way so that you can easily understand. So the surface of our body, right? So which has so many number of proteins or the receptors. Okay, so the incoming bacteria or the viruses, the pathogen, the disease causing bacteria or the virus, first what they should do is they should come and attach to the receptor to get access, to get enter into the body. But unfortunately, because of the presence of the flora, right? So they already occupy those receptors. So because of that, the new bacteria or the virus, which will not have access for those receptors, they cannot go and bind to those surface receptors, so they cannot easily enter into the inside of the body. So that is considered as another mechanical barrier, the presence of the bacterial flora or some other helpful microorganism that is attached to the surfaces, right? So also I mentioned that is why we should not take broad spectrum antibiotics very frequently. That is not good, right? So because if you take broad spectrum antibiotic, all this flora, right? So good bacterias will be wiped. So the opportunistic bacteria or the pathogen, they can easily get access to those receptors and they can easily get enter into the body, right? So one last type of mechanical barrier which I discussed in the previous classes, like acidity or the pH, right? So this is another important thing. Our body does not contain the same pH throughout the body. Different part of the body, we have different pH. So in some places we have very acidic pH, some places we have neutral pH, some places we have the basic pH, right? So our body is containing different types of fluids of different pH, right? So this pH plays very important role for preventing the entry of the bacteria or the virus, because the highly acidic pHs, they're not good for the bacteria or the viruses. They themselves can kill the bacteria or the virus. They can prevent the entry of the bacteria very surface level itself, right? So whatever I have discussed here so far, right? So skin, mucus, the lung, pulmonary surface, cilia, and the flora, the pH, right? So all these are kind of mechanical barrier for the entry of the pathogen from outside the body to inside of the body. Is it understandable?
Yes, sir.
Okay, so Aishwarya, I have repeated this only for you, right? So you said that you disconnected in the last class. So I hope you understand. This is what exactly we discussed in the previous class. So let me ask you a question: whether all this comes under innate immunity or adaptive immunity? Just now I
Innate,
defined,
sir.
right? So yeah, it is innate immunity, right? So all these are kind of innate immunity. All the mechanical barriers are innate immunity, right? So the main job, the main objective, the main purpose of the mechanical barrier is somehow preventing the bacteria or the virus to enter into the body, right? But we cannot say that always the our body will succeed in this process. There are some cases in which, even though we have so many mechanical barrier, there are chances that some of the bacteria or the viruses, um, okay, so let me take this, some of the bacterias or the viruses, they can somehow get enter into the body, right? Okay, so they can somehow get enter into the body, they can clear the mechanical barrier, mechanical protection by the body, right? So they can enter into the body. That is the objective of those bacteria or the viruses, right? So once if they enter into the body, they will be very happy, right? Because that is what they need, literally, right? So they wanted to use the machinery of the host to undergo the replication and all, right? Somehow they breach this mechanical barrier by the skin or mucous layer or the presence of the cilia or the flora which is present or the pH, right? Somehow they enter, right? So which means that they are very stronger. Okay, the bacteria is stronger comparing to your immune system, or maybe some other reason, right? So the strength of the bacteria is very large, or somehow the barrier is breached by the bacteria or the virus. So now they entered inside the body. Okay, so what should happen now? I just make it as an open question. So literally this bacteria are very happy, right? Is our body happy now?
No, sir.
No. Why? Because why the body is not happy? Because we don't need any disease causing pathogen to get enter into the body, because they will disrupt the normal functioning of the cell. Am I right?
Yes, sir.
They will collapse the cellular machinery, they will change the signaling cascade which is normally taking place. So that should not happen. So our body should somehow tackle this, right? Okay, so the point is that once if the bacteria entered into the body, the first thing what they will try to do is that they will try to hide. Because bacteria, virus, any other pathogen, they're very clever, right? So they can understand that if it is visible to the body, okay, so the body will try to kill. It's a natural thing, right? So that is how everything is evolved. So what it will try is, once if it entered into the body, immediately this bacteria or the viruses or any other pathogen, they will try to hide. Okay, they will go and try to sit somewhere, some place where the body cannot easily identify the presence of the bacteria or the virus, right? So what they can do is that this bacteria or the virus, they can quickly try to cross the blood brain barrier. It can reach the CNS. So once if it enter into the CNS, the body cannot easily detect the presence of the bacteria or the virus. Or it can hide in some other tissues, muscles. It can go and sit inside some muscles or some other tissue where our body cannot easily locate or find the presence of the foreign element. Or it can go and sit somewhere in the cell where the body cannot easily recognize or identify. So the main job of this bacteria and the virus, it's going to be hiding themselves from the host. Okay, that's what's the main job after entering into the body for the pathogen microorganism. So just like that, our body is made in such a way that even if they try to hide themselves, we have some mechanism to locate them, find them. Our body has some mechanism to find the presence of those pathogens within the body. Okay, so it's a very beautiful scenario, right? So one is trying to hide, the another one is trying to find out. It's like our body does the job of police, right? So what should happen now? Our body should react, right? Our body should react against the pathogen. This is what we expect, and that should happen, right? The question of interest is that how that is going to happen. Okay, so let's say that let me slightly modify this. Okay, so how this is happening, that's what I'm going to explain in this lecture, right? So because understanding of this is very important for before learning about very minute things, right? So I'm just going to give the overall picture. So our body has some cell or cells, types of cells, which are present very close to this surface level. Okay, so this is the way where the viruses or the bacteria, they breach, they somehow trying to enter, right? So very close to this behind, just after this we have different types of molecules. Okay, so let me say we have different types of cells. We will see in detail, right? So what are those cells? Just understand that we have many different types of cells which are very close to the surface, right? I'm not going to teach very detailed, right? So just what is what, right? So what are the cells? So we can say, for example, we have a cell called as dendritic cells or dendrites. Dendritic cells or the dendrites. So we have macrophage. So all these are present very close to this surface, right? So these are the cells which are present very close to the surface. So let me sketch the dendritic cells. So they will be having dendrites, right? So something like this. Let me say this is the dendritic cell. Okay, so macrophages are kind of very simple thing. So let me say you have to assume, right? So this is the macrophage. So these are the kind of cells which are present very close to the surface. So once if the bacteria or any other microorganism which entered into the body, so immediately these cells which are present very close to the surface, okay, so they comes into picture, they identify, okay, so they can recognize that there is a bacteria which entered into the body. Okay, so it has to do something, for this, right? So otherwise it will disturb the entire cellular system, right? So what it actually does is that it is going to trap, okay, either this dendritic cells or this macrophage, can be any cells, right? Many cells there, right? So it should trap. Okay, so it should trap. Okay, so once if the bacteria or the viruses which is trapped by this dendritic cells, right? So it cannot move to some other part of the body, cannot cause any harmful thing, or it cannot do its job. Okay, the bacteria or the virus, it cannot do its normal job. So it will be trapped by any of the phagocytes, like dendritic cell or macrophages. Those are called as phagocytes. So can you tell what are phagocytes? What's the meaning of the term phagocyte? So I'm looking for some kind of response. I
They
think
protect
you completed
body,
your.
sir.
No,
They
I'm
protect the
not.
body
They
by
obviously. No, what is the literal meaning of phagos? Okay,
killing it.
phagocytosis. Okay, they're going to kill. Okay, anyway, right? So some way they're going to kill, right? So the some way going to kill the bacteria which entered into the body, right? So that's the main job. So it is not only because of the dendrites or macrophages. No, we'll see the details in some other session, right? So there are so many cells which are a part of this. So just for example, dendritic cells or macrophage, right? So those are very common. That's why I have given. So they're going to trap this. So let me draw this. What's the meaning of, we take macrophage here. So say, for example, the macrophage is going to trap, phagocyte this. Phagocytosis is the process by which engulfing will take place. So what it does, it engulf this bacteria. Okay. So engulf this bacteria. Again, the bacteria is not going to be happy here because, it is trapped. Okay, by the phagocyte. It's not so happy. So if we look at uh, uh, the architecture of this cell, okay, so this cells will be having a vacuole. Okay, so let me draw this as the vacuole, and this vacuole will have many number of chemicals, okay, which are actually harmful chemicals. Vacuoles are the fluids with some chemicals, right? So what happened is that the bacteria or the virus which entered into the body is now trapped by some of the phagocytes like dendritic cells or macrophages, which are really present right near the surface. They grab the pathogen and they eat it up. Okay, so the main job of phagocytes are eating, right? So they eat the pathogen. So that is called as phagocytized, right? So there's a term called as, so this is uh, PHA GO, phago, CYTIZED. I think I am right. Okay. So eaten up, right? So it is taken inside the cell. Now the pathogen is present inside the cell. It gone inside the cell. So this cell already has some vacuole. Okay. So this is also present inside the vacuole. Actually, it should be present inside the vacuole. So this has some chemical, right? So what should happen now? This cell should kill this bacteria or the virus, right? So what it does is that kind of fusion, right? So this two vacuoles will fuse. Okay, so this two vacuoles will fuse in such a way that the chemical substances which are present in this vacuole can spill into this over the bacteria, and because of that the bacteria will be broken into pieces. Is it understandable? I think this is a very normal thing you might already studied. Is it understandable?
Yes, sir.
Okay, so let me take this as again. Okay, I'm just going to modify this. The next step of this process is just again, I repeat, just we are seeing the big picture, right? So what's actually uh, yeah. So here, so we have the vesicle here with chemical. Now both of them are fused, right? So because of that, so this bacteria is now broken into many pieces. So say these are the pieces of the bacteria. Okay, it's completely, bacteria is broken into many pieces. So what is the next step? The next step is that this macrophage or the dendritic cell, they are called as antigen presenting cells. Okay, so I'll explain that later, right? So just for understanding purpose, so they will be having a receptor, a protein molecule. Okay, so say this is called as MH MHC2. Okay, so the meaning of MHC is major histocompatibility complex 2. At this point, no need to worry about that. What is that? What is its job? It's a surface protein. Okay, so it's a surface protein. So after destroying the bacteria, this cell, what it does is that it take some pieces of that and put it on the present it to this surface protein molecule called as MHC2. Those red color spots, right? So those are the component of the pathogen, bacteria or the virus. Is it understandable? So what's the main job of MHC2, major histocompatibility protein 2? So it's basically a protein molecule used to stick part of pathogen to the outer surface. So can we call this as antigen? Okay, just think of it, right? So we'll discuss that in detail. So this is so far, is it understandable? But we have to answer so many questions, right? So we have to answer in the upcoming session. We have to answer so many questions, like how the phagocytosis occur. Okay, just I'm giving the big picture in which I did not tell how the phagocytosis occur, how the fusion occur. We don't know, right? So we have to discuss in detail how the lysis of the pathogen it's happening. So and we are saying that MHC, okay, MHC2. Okay, so how it is manufactured? These are very interesting questions we have to answer while we move on during the course. General ways of formation of MHC, right? So these are things we have to address, right? So we'll see that later, right? So as of now, what we understand is that the small pieces of the bacteria or the virus which are presented by the MHC protein which is present in the outer surface. Is it understandable?
Yes.
Okay. So along with that, okay, another thing is also happening here. So it is presented. Okay, good. So MHC, so the pieces of the bacteria is presented in the surface protein molecule. So one more thing is also happening here. Okay, which is that now this macrophage, this cell, is perfectly ready for secreting a type of chemical compound which is called as IL-12. Okay, IL stands for interleukin. Again, I repeat. So it's a antigen presenting cell, so which presented the antigen via MHC2. Immediately, it has now the ability to secrete a new chemical compound which is called IL-12, interleukin 12. So we have different types of interleukins like IL-1, we have IL-6, we have IL-8, we have IL-12, and many more. All right, many more. As of now, this system, this cell is now have the ability for secreting a chemical compound which is called as IL-12. Is it understandable?
Yes, sir.
Okay. So okay. So now we are coming to the very important part of the immune system. If you understand this so far, I'll be happy, right? So because this understanding is very important for what I'm going to out. Maybe I'll draw it again. Okay, I'm not able to copy that. So where we are now is that, so we know that what is this? So here bacteria is broken into different pieces, and some part of that bacteria is presented via protein molecule called as MHC2, because macrophages, these are antigen presenting cells, right? So this is so far what we understand. So also now this molecule, this cell is capable of synthesizing another molecule called as IL-12, which is called as interleukin 12. This is our understanding so far. So the another job of this cell is now that, it's a very important part, right? So because whatever I explained so far is a part of innate immunity, right? So we have another type of immunity called as adaptive immunity. So if you attended my previous lecture, I told that there is a connection between the innate immunity and the adaptive immunity. There is a mutual understanding between the innate immunity and the adaptive immunity of our body. Okay, so that is what's going to establish here, right? So at this stage, right? So this okay, so let me put this slightly down. Okay, so this will avoid the confusion. So now this cell is ready to run, okay, to some other part of the body. Okay, so I told that this is present near the surface, right? Now this cell is ready to move to some other part of the body. So it will start running to some other part of the body. So where it runs, so that place is called as lymph node. So now it presented the antigen, it's having the antigen on the surface, right? So it is running to a place in our body called as lymph node. Okay, what is present in the lymph node? So lymph node is the place where we have the lymphocytes. Okay, so lymphocytes are basically present in the lymph cell. Say for example, the T cell, the B cell. Okay, so these are called as lymphocytes, which are present in the lymph node. So straight away, this cell is now running to the lymph node uh, to show that it captured some antigen, some bacteria has entered into the body, some virus has entered into the body. It phagocyzed it, and these are the small pieces of those bacteria or the virus. So it is going to present that information to the different cells which are present in the lymph node called as a T cell and B cell. Is it understandable?
Yes, sir.
Okay, so once if this is presented to the T cell or the B cell, so the T cell and B cell, okay, so they are the part of acquired immune system. They're not part of innate immune system. They're the part of acquired immune system. Now you can understand this presentation to the T cell and B cell is very crucial for enabling of the acquired immune system. Otherwise, the acquired immune system will not be enabled. So once if this information is presented to the T cells and B cell, then only the acquired immune system will be enabled. Otherwise, it won't be enabled. Is it understandable? So now we can say
Yes,
that
sir.
there is a connection between the innate immune system and the adaptive immune system. Also, there is a reverse connection between the acquired and the innate. I'll explain that, right? So as of now, I think you have understand. Am I right? I'm looking for some kind of response.
Yes, sir.
Okay. Okay, so let's move on to the next part, right? So which is very important. So I'll be talking about um, so I'll be talking exactly about a concept called as IL-12 gamma interferon access, right? So slowly we'll try to understand what is it, right? So now we are in the stage to understand that the connection between the innate immune system and the acquired immune system. Okay. So so can you tell how this IL-12 is synthesized? How the IL-12 is secreted? So here, right? So once if the phagocytosis is over, right? So once if the feces is presented to the MHC2, immediately the IL-12 start secreted by this macrophage. So this IL-12 is going to make the bridge between the innate and the acquired arm. So this IL-12, right? So this cell literally what it does is that it go to the acquired arm, one of the major arm of the immune system, where as I told that we have some lymphocytes like T cell and B cell. So let me draw the T cell, right? So it is going to present this to directly to the T cell. Okay, so let us assume that this is a T cell, or this is called as T0 cell. So let me label this as T0 cell. So normally this T cell is used to be present in the thymus. We will discuss those later, right? So this is called as this T0 cell. This is actually a helper cell. Okay, T0 helper cell. After a couple of minutes, you can yourself understand why it is called as a helper cell. So this cell is called as naive T cell. So the literal meaning of naive, okay, the meaning of naive is that it is not matured, or it is not subjected to the situation already, or there are many terms by which we can which is present in the lymph node. Okay, so this naive T cell, simple way to understand is that it don't have any idea about what is its job. Okay, within the body, right? So it's a kind of, it is not matured, which does not know what is its real function or the job. But interestingly, this naive helper cell, T cell, T0 helper cell, which has so many receptors on it. Okay, there will be many receptors on this T0 helper cell or naive T cell, right? So what happens, this IL-12, okay, so this IL-12, it is now ready to go and attach to this receptor. I'm writing here, IL-12 is attached to this receptor. There are so many receptors here, IL-12. Okay, the IL-12, it can go and bind to this T0 cell, naive T cell, and it can convert this naive T0 cell into other type of cell called as so now this knows that what is its function. Okay, because of the binding of that IL-12, it has some idea that what it should do now, what's the purpose of it's present inside the body. So now because of this, this T0 cell is now converted into T1 cell. Is it understandable? The T0 cell is converted into T1 cell. So how that conversion, how it is converted? Because of the binding of the IL-12. So from where the IL-12 came from, the IL-12 is released by, it is secreted by the macrophage or some other antigen presenting cell. Is it understandable? Sorry?
Yes, sir.
Somebody is speaking. Yeah, somebody is speaking, but it's not audible. Okay, I hope that you people understand, right? So now this T1 cell is very smart comparing to it's no more naive cell. Okay, because it know that what is its job. It's a very smart and happy T1 cell. So what it does is that this T1 cell is now ready to secrete another chemical compound called as IL-2. I give a couple of seconds or minute for you. Just go through this, whatever we have discussed. So let me zoom out. Okay, is my screen visible?
Yes, sir.
Yeah, once you just go through, right? So digest the simple concepts, right? So even though this is simple, this is very important. Is it fine? Once, if you're okay up to this, I can go to the next stuff. So which cell releases IL-2?
T1 cell.
Which cell releases IL-12? Which cell releases IL-12? Which
Antigen
cell
presenting
secretes
cell.
IL-12? And what is meant by antigen presenting cell? Why it is called as antigen presenting cell?
It exposes the broken bacteria to the surface of the cell.
Then
After which IL-12
it
is
is
released
presenting
and
those antigens. Yeah, proceed.
it exposes the broken bacteria after phagocytosis to the MHC2, after which the IL-12 is released, which goes to the lymph nodes.
Okay, which goes and presented to the lymphocytes. Okay, T cell. Okay, so those we will discuss later, right? So as of now, okay. So the beauty is that here there is a connection between the innate and the acquired immune system. So this T1 is a part of innate or acquired immune system.
Acquired cell.
Can I repeat my question? Okay, so this is a part of acquired immune system, right? See here, the acquired immune system is activated by the innate immune system because of the release of this IL-12. Hope I am not confusing you. Okay, so in return, this acquired immune system is going to activate the innate immune system. So how it's going to happen? So this now T1 cell, okay, which is capable of secreting another type of molecule called as alpha, sorry, not alpha, it's gamma interferon. So let me erase this. Yeah. So this is, it is going to release another molecule. It is going to secrete another molecule called as gamma interferon. Okay, so now the question is, what are the two type of molecules which are produced by T1 cell? One is the IL-2, another one is the gamma interferon. Okay, so this gamma interferon, okay, what it does, it goes back, very important. Okay, if you want to understand the concept clearly, just follow me. It goes back from the acquired immune system to the innate immune system. So macrophage is a part of innate immune system. The interferon, which is a part of acquired immune system, which goes back to the part of innate immune system, macrophage, where this gamma interferon help this macrophage or the cells which are participating in this process. So it enhances the activity, speeding up the activity, accelerating the activity of this macrophage. So in other words, we can say that this gamma interferon, because of the presence of this gamma interferon, okay, more and more number of the pathogens which are present inside the body can be killed by the macrophage. The job of the macrophage will be accelerated to great extent because of the release of this gamma interferon. Very important. This is how the innate immune system is enabling the acquired immune system, and in return, how the acquired immune system is supporting the innate immune system. Is it understandable?
Yes, sir.
Okay, so this is what I mentioned as here. This axis is what is called as IL-12 gamma interferon axis. Is it understandable? Very important for examination. IL-12 gamma interferon axis. You have to adjust with my handwriting. Okay. Yeah. Are you able to understand this?
Yes, sir.
Yes, sir.
Okay, so I'm going to slightly confusing you now that this T0, okay, naive helper T cell, I told that it can bind, it has many receptor, right? So let me remove one or two receptor here. Let me make one or two receptors free. I'm not removing. Okay. So let's say a few more receptors there, which are not occupied by IL-12. So I told that T0 cell which can bind IL-12, and binding of that IL-12 convert the naive T cell into somewhat smart T cell called as T1 cell. So along with this IL-2, there's another chemical called as IL-4. Don't get confused, right? So not confusing you. There's another interleukin called as IL-4. So this IL-0, it can also bind this IL-4. Does it make sense? So far, what are the two interleukins which can get binded to T0 cell? One is IL-12, another one is IL-4. Is it understandable? I'm looking for kind of response.
Yes, sir.
Yes, sir.
Okay. So now we just move on further, right? So now here where we stopped is like, so this guy produces IL-2, right? So what is the job of this IL-2? We have to see. So let me draw this, right? So who produces IL-2? So IL-2 is produced by this T1 cell. Okay, so say this is a T1 cell which produces the IL-2, which secretes more and more number of IL-2. So what is the job of this production of this IL-2, right? Um, so there is another type of cell in the body which is called as, let me draw this as maybe green color. Okay, so this cell is called as cytotoxic T cell. I just make this representation. Okay, so this its role is very important, right? So it has a very important role in the immune system. So this is called as cytotoxic T cell. Okay, so what is the main job of this cytotoxic T cell, right? So in the beginning, if you remember, I told that whenever the bacteria or the virus which enter into the body, which will try to position itself in a hidden place where the body cannot easily find out, locate it. Say for example, the pathogen is entered into uh, nucleated normal cells, right? So say this is the pathogen which is entered into the cell. So obviously, whenever any pathogen which entered into a cell that is not good for the cell, we can say in other words, this cell is now a kind of infected cell. Okay, because this pathogen will not allow the cell to perform its normal function. A lot of malfunctioning will be going on inside this. So what should happen? This is now present inside the cell, right? So what is the main job of this cytotoxic T cell? So this cytotoxic T cell which receive this IL-2, okay, which receives this IL-2. So once if the IL-2 is received by this cytotoxic T cell, okay, so what it can do is that it can easily recognize, okay, what are the cells which are infected. Again I repeat, so this now can recognize what are the cells. So say this is a normal cell, this is the infected cell. So it can easily distinguish between the normal cell and the infected cell. Okay, so it can easily recognize that this is the infected cell. So how it is able to recognize that this is the infected cell? That is because of presence of another type of major histocompatibility MHC type called as MHC1. Okay, so it look for the presence of the MHC1 level of the MHC1. So based on that, it detect whether it is a normal cell or it is a infected cell. And if it is an infected cell, if it recognize, if the cytotoxic T cell recognize this as an infected cell, then immediately this guy cytotoxic T cell, it kill the infected cell. Does it make sense? So let me start from there. So let me tell what is the difference between this MHC1 and MHC2. Not the difference. We will see the difference later, right? So what type of cells? Okay, what type of cells that can produce this? That can produce and use this MHC1, and what type of molecule which can produce and use this MHC2? So before that, what is meant by MHC? So many times in my previous lecture, I have expanded the MHC. It is called as
Major histocompatibility.
Aishwarya. So MHC, it stands for major.
Sir,
So somebody can unmute and you
major
can tell what is
histocom
major histo go slow.
major
Compatibility
histocompatibility complex.
complex 1
Complex.
and major histocompatibility to complex 2, right? So that is MHC stands for. It stands for major histocompatibility complex 1 or 2, right? So what type of cells they will be having this MHC1? Okay, so our body has different types of cells. Okay, a series of cells which are called as nucleated cells. Okay, any nucleated cells, okay, they should have nucleus. Any nucleated cell, they can have this molecule called as MHC1, major histocompatibility complex 1. So already we discussed that what type of molecule they can have MHC2. So let me go to that area, right? So what type of molecule is this? MHC2. This is not nucleated. So here while discussing, we discussed about the dendritic cells or macrophages, right? So those are the cells which can have this MHC2 type 2 type. Okay. Right, so that is very important. So moving on further, right? So yeah, so let me put some graphical abstract here. Say for example, there is another type of cell, okay, so which is completely hided or masked. Okay, so which is completely masked, so we can call this cell as unknown cell. Is my voice clearly audible?
Yes, sir.
Okay, so this is called as unknown cell. Okay, literally speaking,
Yes,
we
sir.
don't know what is that cell. Okay, so there's another cell called as unknown cell. So this unknown cell, it is capable of producing a type of molecule called as IL-4, interleukin 4. So as I said in my previous lecture, we have different types of interleukin. So this unknown cell, this is capable of producing a type of interleukin molecule called as IL-4. So what's the main job of this IL-4? It's very simple. So already we discussed about T0 cell. If you remember in my previous lecture, I talked a few words related to one of the very important cell of the immune system called as T0. Okay, so say for example, this is the T0 cell. So this T0 cell is having many number of receptors on the surface. So some molecules can go and bind to this surface, and the binding of this IL-4 to this receptor on the T0, which literally convert this T0 into another type of cell, which is called as T2 cell. Okay, so T2 cell. So now this T2 cell, interestingly, this is capable of producing another few types of molecules called as interleukin 5, IL-5, and IL-4, interleukin 4. Does it make sense? Looking for kind of response because of the continuation of the previous lecture. So this is a T helper 0 cell, which is converted into T helper 2 cell, which is capable of producing two types of interleukin called as IL-4 and IL-5. So previously we discussed how the T0 is converted into T1 while discussing about the IL-12 gamma interferon axis. So now I am introducing different concept that how the T0 is converted into another type of cell called as a T2, which is capable of producing IL-4 and IL-5. Does it make sense?
Yes, sir.
Yes, sir.
Okay, so what is the name for this T0 cell? If you attended my previous lecture, we call this as naive cell. Okay, so which is not having prior experience, which is not previously exposed, which does not know what is its role in the human immune system, right? So once if it binds the IL-4, okay, so it is matured, it is converted into the T2 cell, which produces IL-5, IL-4, okay, which can do much better thing for doing something against the pathogen which are present inside the body. That is what I'm going to explain now. Okay, so we just start with this IL-4 and IL-5. With this part I am going to discuss, right? So this part, what is happening here after the conversion of so let me make a copy of this. Okay, so I just made a copy of this. It will be easy for discussion, right? So now, as of now, the naive T0 cell is converted into T2 cell. It is, secreting. It started secreting the IL-4 and IL-5. Okay, so this is called as a T helper 2 cell. So what is the role of this T helper 2 cell? So it plays a very important role in immune system, right? So very important for examination, right? So it has very important role in the immune system. So what it does is it is going to act on some of the component of our body, some of the other cells which are present in our body. So let me draw this as a B cell. Okay, so I'm going to talk about the B cell. First, I am talking about the B cell, right? So let me put this as a graphical illustration. So say this is the B cell. Okay, so let's make it as a B cell, right? Say this is the B cell. So this IL-5, IL-4, which are produced by this T1, this system is now capable of acting on this B cell. It's going to act on this B cell. Okay, B cell is one of the major component of adaptive immune system. We'll see that in detail, right? So what is the specialty of this B cell? The specialty of this B cell is it is a kind of weapon factory. Okay, so it has so many weapons. It can produce so many weapons, which can be used for killing the pathogens which are present within the body. Okay, so in generally any textbook if you take, they refer this as the weapon factory, which can produce many different types of weapons. So if we say okay, so because of the interaction with IL-5, IL-4, so this B cell, okay, which which is getting matured further. Okay, so let me put something like this. So we'll give some variation. So it undergoes some kind of change. Okay, so in which what happens, so it started producing some molecules within it. The cell will start producing some molecule after listening to this IL-5 or the IL-4. So they can activate this B cell. They can activate this B cell, so which started to produce some kind of chemicals within the cell. Those chemicals which are called as immunoglobulins, or we can say antibodies. Very important. Okay, so the main job of this B cell is, okay, so whenever the pathogen enter, right, so the antigen is presented to the cell. So this is the cell. B cell is the specific type of cell which is capable of producing large number of antibodies. Okay, so once if they produces large number of antibodies, so this B cell is free to move everywhere inside the body. Okay, so wherever the blood is flowing, this B cell also can flow. Throughout the body, this can go, and it will start fire the antibodies which are produced by this. So these antibodies are very specific for the antigens which are presented to this B cell. Okay, so what happens that throughout the body, it'll start firing this antibody. So these antibodies will go and act on the pathogen, and it can kill the pathogen.